All right. Well, welcome to OA Week 2025, everyone. We'll give people a few moments to trickle in and we'll get started in just a minute. Okay, welcome everyone, and thank you for joining us for OA Week 2025. My name is Carly LaRoche, and I'm a member of the GOA-ON Secretariat. Please note that this session is being recorded. OA Week is presented by 4 sponsoring organizations. First, GOA-ON, the Global Ocean Acidification Observing Network. Second, NOAA, the United States National Oceanic and Atmospheric Administration. Third, the IAEA OA-ICC, the International Atomic Energy Agency Ocean Acidification International Coordination Center. And last but not least, the IOC, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization, or UNESCO. GOA-ON was established in 2012 with just a handful of members. Since then, it has grown immensely and now has over 1,200 members from over 110 countries. GOA-ON also consists of 11 regional hubs, which span across continents and oceanographic regions. We'll be hearing from most of them throughout OA Week, and we're lucky enough to be in a hub session right now for the North American hub. If you're not a member yet of GOA-ON, you can join today by visiting goaon.org, and I'll drop information in the chat about how to join. OA Week debuted in 2020 and returned in 2021, when events and conferences were postponed due to COVID-19. Following the successful in-person symposium on the ocean in a high CO2 world in 2022, OA Week has become an annual event to maintain momentum around OA research and provide a platform virtually for the ocean acidification community to exchange their latest findings. We're thrilled to present a wide range of ocean acidification topics and speakers from around the world throughout OA Week. A few notes on housekeeping before we get started. During the presentations, all participants are in listen-only mode. You're welcome to type any questions you have into the questions box, which can be found at the bottom of the control panel on the right-hand side of your screen. We'll be monitoring for incoming questions and we'll pose them to our speakers during the question and answer period after their talks. For the discussion, you can also use the raise hand function in the toolbox at the bottom of your screen, and we can call on you to ask you your question directly during that question and answer period. And with that, I would like to introduce you to your moderator for this session, Dr. Helen Gurney-Smith. Dr. Helen Gurney-Smith is a federal research scientist with Fisheries and Oceans Canada, where her group works on the impacts of climate change on marine species and ecosystems. She's the new chair of the North American Hub and has worked on ocean acidification biological impacts for a number of years, and more recently on MCDR ecosystem impacts. Helen, I'll hand it over to you.
Hi, thank you so much for the introduction, and to everybody for tuning in wherever you are. So good morning, good afternoon, good evening. So this session is going to be for the North American Hub session, and this hub is one of the earliest formed hubs within GOA-ON, and it includes over 225 members from Canada, the US, and Mexico. But absolutely everybody is welcome to join us, so please do. And there's some different resources and activities linked on the slide to give you a bit of a taste of the kind of work that we do. The GOA-ON North American Hub has been integral to GOA-ON, with the members serving not only within the hub, but also as GOA-ON Co-Chairs and Secretariat members. The theme of this week's GOA-ON OA Week sessions in 2025 is Rising to the Challenge of Ocean Acidification, so using science and knowledge to drive action. And this year's session for the North American Hub will focus on new information on OA science, knowledge mobilization and action from Canada, Mexico, and the US. Each speaker is going to give us a 15-minute presentation, and then we'll hold all questions for the end, where we'll have a 30-minute discussion as a group. And with that, we'll begin with our first speaker. So I'd like to introduce Dr. Kristina Barclay, who'll be presenting on the state of Canada's ocean acidification knowledge. Dr. Kristina Barclay has a background in conservation paleobiology, ocean acidification, marine invertebrate biology, and also science communication. She's been helping to build the Canadian ocean acidification community of practice since 2020 and has recently joined the GOA-ON North American Hub Steering Committee. So a big warm welcome in that role too. She's leading a community perspectives paper on the state of Canada's OA knowledge and recommendations for needed actions to address ocean acidification in Canada. And with that, I'll pass it over to you, Kristina.
Amazing. Thank you so much for the kind introduction, Helen. And I'll just start sharing my screen. That look good?
Looks good.
Yep.
All right. Yeah, so thank you so much, Helen, for that introduction, and Carly and everyone, all the organizers of OA Week. This has just been such an exciting week, and I'm really honored and thankful to be presenting here today. So today I'm speaking from Victoria on Vancouver Island in British Columbia. And so I'd just like to acknowledge with respect the Lekwungen speaking peoples on whose traditional territory I live and work, as well as the Songhees, Esquimalt, and Wasanich peoples whose historical relationships with the land continue to this day. And so if you live in someplace like North America, I'd encourage you to take a moment to think and learn about the lands that you might work on and the traditional caretakers of this land. So today I'm going to give a really high level overview on some of the OA work and some of the efforts that are occurring across Canada, and as well as a snapshot of the current state of Canada's ocean acidification knowledge. And I'd just like to take a moment to acknowledge this has really been a community-driven effort, and we've had so much help and support from our community members and some amazing co-authors that have really helped shape these efforts. So thank you so much to our community. So in keeping with the theme of OA Week this year, I'd like to acknowledge that these are just the first steps in pushing forward the needle on OA science to action in Canada. So for some context, I've included this figure from Duke et al., and it shows a map of Canada with major ocean currents and ice cover. So Canada is uniquely vulnerable to ocean acidification for a number of reasons. We have the longest coastline of any country in the world. We're also surrounded by 3 ocean basins that each have unique processes that make them susceptible to OA. And we're also situated at high latitudes, which increases our natural vulnerability to OA given the lower solubility of carbonates in colder waters. In the Pacific region, there are naturally corrosive conditions due to circulation patterns of old and cold seawater, and that's exacerbated by seasonal conditions like upwelling. In the Arctic, accelerated melting of sea ice as well as freshwater input increase carbon dioxide absorption, which leads to accelerated ocean acidification. In the Atlantic, ocean acidification is exacerbated by the input of acidic waters from the Arctic, as well as the Labrador Sea and the St. Lawrence River here. These many drivers and unique regional conditions really pose a large challenge when it comes to coordinating responses and adaptation strategies across a country as large and relatively sparsely populated as Canada. And the needs and priorities of vulnerable communities and sectors are different between regions. So for example, along the west coast of North America, the impacts of ocean acidification are already being felt by the oyster industry, which has prompted a lot of responses. Whereas on the Atlantic side, more of the focus is on socioeconomic fisheries like lobsters, for example. And in the Arctic region, subsistence species, impacts to traditional ways of living, and connections to resources are all major concerns. And also across the country in general, subsistence and traditional food sources are important protected rights for Indigenous communities, but these vary regionally and vary in terms of ocean acidification impacts. So in an effort to try and connect the OA community across Canada, the Canadian Ocean Acidification Community of Practice, or the OA-COP as we like to call it, was founded in 2018, sorry. And this community of practice is funded and supported by MEOPAR, which is the Canadian Marine Environmental Observation, Prediction, and Response Network. And it's led and supported by, the community of practice is led and supported by the Tula Foundation, as well as Fisheries and Oceans Canada, or DFO. We're also a UN Decade Ocean endorsed project under the OARS program. And our overarching goals are to coordinate across sectors, disciplines, and regions to share OA expertise and data and increase OA literacy and resource development, to identify pressing needs for OA research and knowledge, and to create a collaborative and supportive environment for groups that are affected by OA. In the first 5 years of our COP, we're primarily focused on building resources and building connections across the country. And now, though, as we look to the future and the second half of the UN Ocean Decade, we've turned our focus towards knowledge translation and implementation of OA actions in Canada. Our COP is led by myself as the coordinator, as well as our two co-leads, Dr. Helen Gurney-Smith, moderating the call here today, and Dr. Wiley Evans, who's at the Hakai Institute. We're also in the process of reforming our steering committee, and we've had the pleasure of working with a lot of really great engaged steering committee members in the past over the years. Our community consists of about 160 members from government, primarily at the federal level, academia, various NPOs, fisheries and aquaculture sectors, as well as community leaders, students, early career, and international members. So one of our major initiatives to date is this synthesis community perspectives paper on the state of ocean acidification knowledge in Canada that's currently in prep with a hopefully near final draft currently being reviewed by our many amazing co-authors. And so I'll spend most of the rest of the talk giving you some really high-level sneak peek results from that paper. We also provide supports for ongoing OA initiatives across Canada, such as the province of British Columbia's Ocean Acidification and Hypoxia Action Plan, which I'll touch on more in a little bit here. We also held a strategic planning meeting in the spring of this year and produced a What We Heard report with 8 major recommendations from our community, and that's now available on our website, so you can feel free to check that out. And speaking of our website, that's kind of our main hub for resources, and we've got a lot of resources that we've developed, including a couple of new databases on OA in Canada experts and publications. So please feel free to check those out and add yourself if you've done work in Canada. Other resources that we have made over the years include species and seafood industry impacts, a map of OA resources in Canada, many blogs and interviews on research papers, newsletters, and more. So please go check out our website, browse our resources, and consider joining our membership. And I'll include a link to that at the end of the presentation as well here. Okay, I'm going to shift gears now and go over some of these preliminary findings from our Community State of Canada OA knowledge paper in prep. So in this paper, we cover monitoring and modeling efforts, biological experiments, as well as socioeconomic and policy perspectives. And then we conclude with the recommendations section that outlines pathways for enhancing OA research and integration of OA knowledge into decision-making, from federal policies to local resource management. In the paper, we also present a map of Canada's OA assets, which includes things like cruise lines, monitoring stations, biological infrastructure and labs, modeling domains, and more. And there's a dynamic version of the map that will eventually end up on our website, and it'll be updated regularly. And also there's going to be an app version for mobile devices, so please stay tuned, and hopefully that'll be up fairly soon here. In terms of monitoring, there's many organizations that contributing to monitoring and observation efforts across Canada, but I'd just like to highlight two of Canada's longest time series that have biogeochemical data, which are long enough to resolve trends and are important contributions to our understanding of OA in Canada. And these are Line P in the Pacific and AR7W in the Central Labrador Sea beginning in the late 80s, early 90s. In terms of modeling, there are 8 regional biogeochemical models that have been developed in Canada, and so you can see their coverage here, although there's a new model that's missing from this figure. And while we recognize that there are several important US-based models that cover parts of Canada, our goals here and for all sections of the paper were to focus on Canada-generated OA data. And then in terms of biological capacity, there's several labs in Pacific and Atlantic regions in Canada that have the capacity to do OA studies. And these are run primarily by DFO or Fisheries and Oceans Canada. But there's also a few facilities that are run by smaller institutions and universities, such as the Hakai Institute's Marna Lab and Bamfield Marine Sciences Center on the West Coast here. In terms of biological responses to OA, it's probably not surprising that the majority of biological studies conducted in Canada focus on socioeconomically important shellfish like crustaceans and bivalves. And it's also probably not surprising that overall most species studied were negatively impacted by OA, although there are a couple of promising recent studies showing resiliency in selectively bred oyster broodstock. So that's potentially important for the aquaculture industry. Another key take home here, though, is that there's still many unknowns. There's still many species that are understudied in Canada. So not only do we not see a lot of negative impacts on important socioeconomic species like lobsters here, which are half of the Canadian seafood industry values, but there's a lot of unknowns for species like snow crab, finfish, scallop, but also important subsistence species like Arctic char and Arctic cod, for example. In terms of OA actions in Canada, the province of BC is really leading the way. So there's both this provincial OA action plan, as well as several examples of local and Indigenous community OA initiatives that are happening across the province. And this was partially due to the impacts to the oyster industry that are already being felt on the West Coast. And it also resulted in ocean acidification being identified as a major risk in the province's preliminary strategic climate risk assessment in 2019. And that was due to the potential financial losses predicted for 2050 for the aquaculture oyster industry. So after the release of this report, there was plans kind of laid for an OA action plan. And so there was a series of 4 workshops held in 2021, 2022 that covered the state of the science, seafood and community perspectives, and policy considerations. And these workshops form the basis of a provincial ocean acidification and hypoxia action plan, as well as a scientific assessment that were released in 2023. From there, the province then provided $1.7 million for a Climate Ready BC seafood program, which funded 11 projects. And these projects are currently underway, investigating, different observation, mitigation, adaptation strategies, and they're planning to wrap up in early 2026. And along with that, we're planning a forum for March of 2026 that will highlight these 11 projects, as well as assess priorities and progress and priorities for next steps for the BC OAH action plan. So if you're interested in learning more about the forum, please check out the webpage here, or feel free to reach out to me, and I'll put my contact information at the end here. So from all this information that we gathered, we then provided recommendations on pathways as well as examples of OA actions that are needed in Canada. And these recommendations were roughly based around the themes of capacity building, research needs, knowledge mobilization, policy action, as well as socioeconomic needs. And the idea here is that the more actions and integration of OA knowledge into policy and planning, the more OA risks decrease and the more societal benefit in Canada would increase. So some examples of capacity building actions that we recommended included increased investment in research and infrastructure for OA work, as well as the support for the creation of a centralized Canadian OA data repository through existing platforms such as CUSE, which is the Canadian Integrated Ocean Observing System. Research needs are many, but we recommend those that focus on filling data gaps for priority regions, species, and models. Knowledge mobilization recommendations were largely focused on building coordinated science advice, as well as communication resources, such as creating an annual State of OA Science report that can be used for informing decision-making. Policy actions are also a major priority, and there's great need to have explicit inclusion of OA in decision-making and policy at all levels. But one example is that we advocate for is the development of a federal OA action plan. And finally, socioeconomic needs focus on linkages between OA knowledge that need to be included in decision-making, resource management, and coastal planning processes, as well as adaptation planning. So basically, in summary, we have a lot of progress that we've made on OA knowledge in Canada and identifying priorities, but there's many gaps that remain, and there's great need to connect our knowledge to decision-making that will ultimately safeguard Canadian ecosystems, communities, and economic sectors against ocean acidification. And above all, we emphasize the importance of emissions reductions as the most important action to reduce OA risks in Canada and beyond. And so with that, I'd just like to take a second to thank our amazing community and co-authors, and thank all of you so much for your time. And please consider joining our OA community of practice.
That's wonderful. Thanks so much for sharing that presentation, Kristina. So just let you know, just a reminder that we will have a question session at the end. So be thinking of the questions that you want to ask, and you can either put them into the chat or ask them directly yourself. But now we're going to move on to the next speaker. So I'd just like to introduce Dr. Leticia Espinosa Carreon, and she'll be sharing her presentation on the advances in the understanding of carbonate system in the Mexican tropical Pacific. So a little bit about Dr. Leticia. She specializes in marine ecology, and she's participated in oceanographic cruises organized by CMAR, ICML UNAM, and IMIPAS, and has contributed to the monitoring of nearly the entire Mexican Pacific. So that's a big region that you're covering there. Her current research focuses on ocean acidification in the Gulf of California and along the western coast of Baja California, particularly from 2016 to 2024, as well as in the tropical Mexican Pacific and the Gulf and in the Gulf of Tehuantepec as well. Okay, I'm going to pass the mic over to you, Leticia.
Hi, everybody. My colleagues' team and I is glad to show you the advances in the understanding of the carbonate system in the Mexican tropical Pacific. Next, please. The Mexican tropical Pacific is a geographic location characterized by unique climatic and oceanographic conditions. In this area, we can find mesoscale processes, high phytoplankton biomass, diverse fisheries, and this is important to say that this is a water tropical pool. And this is a oxygen minimum zone, and this is a influence for the El Niño Southern Oscillation. Next, please. So, what do we know about the carbonate system in the tropical Pacific Ocean of the coast of Mexico? Next, please. We can found three important contribution from the Franco in the left side, in the middle side the Chapa Valcorta, and the right side the Truco Pignata. But this is a few contribution in these zones. Please, next, please. This is important to emphasize the need to increase studies of the carbonate system in the Mexican tropical Pacific. Next, please. The study represents a valuable opportunity to explore the relationship between physical forcing and the carbonite system in the Mexican tropical Pacific. In this study, we aim to quantify the relative contribution of the hydrographic structure and surface thermal forcing to variation in the total alkalinity, DIC, and pH, and to examine how the latitudinal distribution influences these parameters in the Mexican tropical Pacific. Next. So in this part of this methodology, we have satellite images from the sea surface temperature from the January 2003 to December 2023, and the download the Oceanic El Niño Index from this link from the same period. Next. So the wind stress and the upwelling index only have this period, January 2017 to December 2019. So it's important the Hofmüller diagrams from the first 1,000 adjacent to the coastal were considered from the sea surface temperature anomaly, wind stress anomaly, and upwelling index. Next, please. So, aboard the research vessel of the Puma from the UNAM, we can participate in three cruises in the 2070, 2080, and 2090. In the left side, you can observe the 5 transects in the Cabo Corrientes, Jalisco, Manzanillo, Colima, Lázaro Cárdenas, and Maruata, Michoacán, and the southern is Acapulco, Guerrero. In each station, we can take a hydrology water column from the surface to the 2,000 meters with the seabeers CTD+. The conservative temperature and absolute salinity calculation was made with the you can observe. Please, next. So, from the carbon oxides, the water samples were collected at the standard depth to determine DIC and total alkalinity, followed with the recommendation with the Dixon in 2007. In the laboratory de productividad primaria y sistema del carbono, we analyzed with the DIC and total alkalinity, followed with this recommendation. Next, please. So, with the result, we can observe this image with the left side, we can observe the CF5 transect, and we can show you the ENOP in the part up, the top is a negative sea surface temperature anomaly, and in front of the Cabo Corrientes, the south in front of Acapulco, we can observe the positive sea surface anomaly. And it's important to observe the 0 degrees line. This is an important transition zone. So, in the top, we can observe the negative anomalies that coincide with the La Niña events for the different years. So, it is important note that in El Niño 2015-2017, how the whole study area is completely with the positive sea surface anomaly. Next, please. So with this information, we can demonstrate can divide the zone with the 3 important parts. One is in front of the Cabo Corrientes, is characterized by the upwelling zone. Is in front of Maruata, is a transitional zone, and the south in the Acapulco, this is a tropical warm pool. Next, please. So when the related the sea surface temperature anomaly with ONI index, we can have a statistical significance. Next, but when they only try to the years, oh my God, when they sorry, Okay, sorry. I moved something. Okay. When they try to relate the 2070, 2090 years, there's no relation significant, and we consider it neutral years. Next, please. With the same characterization of the division of the area, we can observe the wind stress anomaly, and we observe the positive and highest values for the wind stress anomaly in from the Cabo Corrientes, and the less wind stress anomaly we found it in the Acapulco zone. Next, please. So the index of Walling confirm the three different zones. We can observe the the Welling index that the characterized in from the Cabo Corrientes with the negative anomalies. In this case, is from the November to May, and in front of the Acapulco, we can observe the few upwelling. Next, please. Next. So, can you return, please? Yes. Trasviña and colleagues is reporting from the coastal welling the January to June, but in this work reported the November to April the principal upwelling. López Sandoval also said that in the current stronger upwelling, they bring colder and rich water that enhance primary productivity. Next, please. So we can observe the conservative temperature and absolute salinity diagrams in the most important contribution for the tropical surface water is in Maruata, Lázaro Cárdenas, and Acapulco. When we can related with the other works, for example, the Cepeda, Morales, and Franco, the most important difference there are in the Gulf of the California water and the California current water. So these differences is because the monitoring in different areas and time. Next, please. Yes. We can observe the left side, the relationship between DIC with the surface temperature. And it is important to say that in middle is we can observe the different water masses and they correlate positive significance in the tropical, the three cruces in the tropical surface water is a minus values, and the high values from the pH in the bottom, the pH. So it is a significant correlation, and we can find the highest values of the pH. When show you the total alkalinity with the potential density, we can observe that the total alkalinity is a minus variability. But next, please. We can observe this point. This is a contribution for the Balsa River. So next. In this part of the bottom, we can observe the ocean acidification and biological contribution. So we can observe the different parts of the data for the 3 years. For example, in the left is the number 1. We can observe the photosynthesis and the CO2 emission characterized with the surface samples. In the second one is 80% of the data is located in this zone with the respiration, and the CO2 is important. And the third one is a bottom sample water with the dissolution there are. So in this case, when I compare it with our data with other works, for example, sometimes is similar, but other one is different. Okay, next, please. In this slide, we can observe in the part left, it is the different years and the different zones, and the total alkalinity IDLC we can found in the Mexican Convergence Zone in the 2070 years. We can find that the highest values for the total alkalinity and DIC, and the lowest values, the pH, with the fluorescence. So in the opposite, can find in the 1917 in the same zone, the Mexican convergence zones. In this case, the DIC we found it in the lowest and the highest pH and the fluorescence. Next, that this is confirmed that the phytoplankton photosynthesis in reducing dissolved CO2 and consequently increase pH. Next, please. So, but the signal, however, may be modulated by the respiration, remineralization, and the physical processes. Next, please. By conclusion, we have the division of the study area is proposed into 3 zones: Whaling Zone, the Mexican Convergence Zone, associated with the 0 degrees sea surface temperature anomaly line, and the tropical warm pool with the positive sea surface temperature anomalies. The effect of the interannual events of the zone and the day significant correlation between sea surface temperature anomaly and the ONI index from the period of 2003-2023. The years 2070, 2080, and 2090 I considered neutral, and the relationship between DIC, total alkalinity pH versus potential density per year and per transect was significant, and the area with the greatest variability in DIC, total alkalinity pH, and fluorescence was the Mexican Convergence Zone. Next, please. This is results from some were part of this contribution that is submitted with the Frontiers in Marine Science. Next, please. And so this is we're working with the other contributions more. Next, please. And this is a sunset of the Mazatlan, Sinaloa. Thank you so much for your attention. Thank you.
Thank you so much for your presentation, Leticia. That was amazing. That sunset at the end was fantastic too. Gives you that nice kind of sense of like connectivity with the ocean. Okay. So we're going to be moving now to our third presenter, and get your questions ready for Leticia as well, because we'll be having a question and answer session just after that. So our final speaker today is Dr. Kate Hewitt, and she'll be presenting on patterns in corrosive water across the Washington and Oregon shelf. Dr. Kate Hewitt is an oceanographer and engineer with a background in science communication. She's an active member of the Coastal Modeling Group at the University of Washington School of Oceanography. And while currently focused on the coastal waters of the Pacific Northwest and Salish Sea, she's worked on understanding ocean acidification and hypoxia across the California current system. Okay, Kate, I'll pass it over to you.
Thank you. I'm just getting my talk ready.
Yep, we can see that. Thanks very much.
Great. Thank you so much, and thank you for that kind introduction. I'm actually going to turn my video off while I share. Today we're going to be talking about patterns in corrosive water across the Washington and Oregon shelf. I'd like to take a minute to recognize Parker McCready, who leads the Coastal Modeling Group, as well as Jan Newton and members of the Coastal Modeling Group and OA Indicator Working Groups who contributed to model development improvements and also discussions around the work that we're going to be talking about today. So when all the data that's going to be presented today is from the live ocean model, and that domain is shown in the white here. But we are just going to be talking about the Washington and Oregon shelf. So while live ocean simulates the coastal waters of British Columbia, Washington, and Oregon in the Salish Sea, we're going to be focusing on that blue area within the rectangle. And throughout this talk, the data that's going to be presented is the data that's above this blue shaded region, which is the zone of the Washington and Oregon shelf that's masked between the 15 and 200 meter depth contours. And we'll either be talking about bottom water on the very bottom, surface water that's above this blue shaded region, or data from the entire water column above the blue region. We use two different thresholds to define corrosive water. So today we're going to be looking at aragonite saturation state under 1 or under 0.5. And we're going to be talking about corrosive water in reference to these two thresholds. And just to come back, this colored map here is showing just a screenshot from August 2021, where live ocean has all the temperature colored in. But live ocean brings up-to-date information. We bring in information on winds and sunshine, ocean condition, tides, rivers, including about 100 wastewater treatment plants. And we use all these data to run a 3D computer simulation of our regional ocean, which includes things like biogeochemistry for phytoplankton, oxygen, and ocean acidification. And we use these data to perform research and ask research questions. But we also provide a 3-day forecast to stakeholders and scientists and the public, and we make that available through the NANUS visualization system. And just noting that while we're going to be presenting a lot of model data today, we could probably go through a talk on how we validate all of the model data with actual observations. So this is just a quick plot of one of the things, visualized one of the ways that we compare model and observations, where you have this map on your right, where all the green dots are different casts and bottle locations, and we compare those with model data. And in times where there's a mismatch between models and observations, we then go through and perform model improvements to update live ocean so that we're, as best as we can, replicating and simulating ocean conditions. So you can imagine that if we took that blue shaded region and we looked at the bottom data from the model, and we took 10 years of data and we looked at the months, winter months, January through March, we could come up with a % time that the bottom water was below 1. And we'd see that the inner shelf had conditions where aragonite saturation state was higher than the outer shelf. And we can kind of get a picture of what the shelf looks like through time if we then go in and add all the other months. So each panel here is showing you a progression: winter, spring, summer, fall. And the thing that should be jumping out to you right away is how red and orange the summer is. So much so that you could take the summer and you could, this is just bottom water, you could take the summer and you could replot it using a lower threshold, whereas the third panel is below 1, the fifth panel is using 0.5. And what you can see is that in the summer, there's more corrosive waters across the shelf in the bottom water, and there's some regions that have worse conditions than others. A different way to look at what the corrosive water looks like across the Pacific Northwest is to take, and I know that this plot is going to be complicated, but I'm going to walk you through it, is to take that same shelf region. So this little map is that same shelf region. And then we just took a strip of the shelf. So we took around 40 meters. So we were looking at what does it look like along 40 meters in the north-south, along the north-south axis. So looking at just bottom water, so just the bottom layer of the model, we have latitude on Y, and then we go across time on X. And so what you can see is that same seasonal progression, and you can just look at it across space. The colors are by threshold, so they're not an even color bar, but they're just colored by different thresholds of aragonite saturation state. And we can do a similar thing for the surface water. So this is a similar plot, top and bottom, where we're looking at the surface layer on top and the bottom layer on bottom. And we're seeing that we're just looking at the seasonal progression of corrosive water presence on the shelf in the surface and bottom layer. Something that you might be wondering when you're seeing this is, okay, if there's a progression in surface and there's a progression in bottom water, what does the rest of the water column look like? Because we would think that the thickness of the corrosive volume, the thickness of corrosive water is really important. And if you're thinking that, me too, and I made you a cartoon to understand how we then look at the volume of corrosive water across the shelf. So thinking through live ocean, and I made this, live ocean has 30 different layers, and there's a bunch of grid points in that blue shaded region. So if we look at just one grid point, we'd have something that looks like this. We'd have 30 layers with a bunch of information, different data types from the model. And from that, we can have our cell area, which gives us an area. And then we can also take the thickness of corrosive waters where its aragonite saturation state is below some threshold. We can also let it stagger, where you can not just have a continuous layer where it's below 1, but you can have different lenses of water where it's over 1 or under 1, and we still get that height of or thickness of corrosive water. So then at every grid point across that blue area, we can find the corrosive volume just by multiplying the area by that depth, which would then allow us to look at the volume, which is taking into consideration how much corrosive water in a volume term is present on the shelf, and we can look at that over time. So if we were to sum all those corrosive volumes of each grid point across this blue region, we would be able to find the percent volume that would be under 1. And what we would see is something like this curve, where as you come into the spring, the corrosive volume increases, comes to a peak in mid-August, and then declines in the winter months as we transition to the downwelling season. From the maps where there was the different, where we looked at surface and bottom, you saw that there was a time component where there was seasonality, but you also saw that there was a lot of spatial variation in the presence of corrosive water in a north-south sense. So to understand the Washington and Oregon shelf, we binned them into 6 north-south, 6 bins, and each bin is 1 degree latitude. So using the shelf mask and then these regions, orient yourself with this map, because this map with the same colors are going to be shown over and over throughout this talk. So we have the Columbia River and the Strait of Juan de Fuca, and then we're 6 regions. In this curve, we're looking at the corrosive shelf volume where it's under 1. And you can imagine that if we go to here, it's 60% corrosive, 60% of the shelf is corrosive. And if 60% is bad, then 40% is good. Why I'm saying that is because when we plotted it, we were looking at available habitat. So here, what we're looking at is the available habitat for each region with the percent corrosive volume. So we're looking at all the years 2014 to 2024, and we have the curves just flipped upside down. So what we see is that there's near 90 to 100% water in the early winter that's over 1, which then declines. So the available habitat that's over 1 declines as we go through the summer. And then there's a dip and it then rebounds. The curve is pinched because we're showing 11 years of data. We can then add in the southern regions, and here in blue. And if you had longer to look at this slide, the first thing that you might notice is that the southern regions have a lot lower values. So they have less habitat available in the summer where aragonite saturation state is over 1. The next thing you might notice is that the time spent in the summer where there's a lot of corrosive water is pretty important. So the thickness of this dip is important. And to try to capture that into all one plot where we can look at what does the shelf look like in terms of corrosive water over time, what we did is we took cumulative corrosive volumes. And so to do that, we took the curves from the last slide. We took the corrosive volume, and this is just running through example for R1. We take the volume of water bound by the maroon shading, and we find where the aragonite saturation state is under 1. And then for every year, we take the cumulative sums. So the volume at day 1 is the January 1st volume. At day 2 is January 1 plus January 2, and so on throughout the rest of the time. And from that, we would get these curves. And this is just showing 2014 to 2023 because it's a lot of colors. I took off the y-axis because it's going to give you a really large kilometer cubed, and it does, I don't have a sense for that, so we convert it. But the point you should get is that over the time we're summing all of our cumulative volumes, and the higher up on this y-axis comes, the larger volume and the more persistent corrosive water was present on the shelf. So 2020 had worse conditions than 2016. To get it to make a little bit more sense than having, we just put it into a cumulative fractional volume. So we took the volume of corrosive water and we divided it by the total volume. And what that allows us to do is to kind of get a better sense of what the shelf was looking like. To make that make more sense, if you had 100% of the water column was under 1 every day, you would be summing 100% every day. So you'd sum 1 every day. So if 100% of the water column was corrosive every day, at the end of the year, it would be 365. We can do that for our 2 different thresholds of 1 and under 0.5. And we can grab the values on day 365 for leap year and non-leap years. And then we can plot that across time. So this is kind of our picture of what the corrosive volume looks like across the shelf for Washington and Oregon, where every bar's color represents the region, and the darker bars represent when we're using the threshold of 1, and the lighter bar when we're using the threshold of 0.5. And what you can see is you can see the spatial variability, and you can also see the interannual variability here. Running through just a quick example to drill home like this kind of complicated plot is that if we looked at R1 in 2018, this red bar is the cumulative fractional volume. So how much, what the volume looked like in region 1 across the entire year of 2018 for using a threshold of under 1. And then the lighter bar is looking at the threshold of under 0.5. We add in the southern regions. And again, you see that the higher the southern regions have a little bit higher numbers. They had more corrosive volume present across the year than the northern regions. But also what jumps out is that when you look at the lower threshold of 0.5, as you approach the Columbia River, your values are increasing. So there's a higher volume and it's more persistent as you approach the Columbia River. And this is a way that we've been trying to characterize and understand what the state of ocean acidification is looking like in the Washington and Oregon shelf. And I'm happy to share data for this and point you towards where live ocean data is, or answer any questions about these plots are what we've done. And my email is down here at the bottom. Thank you.
Thank you so much, Kate. That's amazing. What a massive volume of work. And I'd just like to say thank you to all of our speakers today for doing such an amazing job and such a diversity of topics too, which has been wonderful to see in different outputs. We'd now like to open the floor to questions from the audience. So you can either type your question into the questions box, which can be found at the bottom of the control panel on the right-hand side of your screen, or you can raise your hand and we'll call on you to ask your question directly. So great news is that it's already sparked some discussion, and we have a couple of questions in the questions and answer box already. So Natalie, you had the first question. Would you prefer that I read it out, or would you like to ask your question directly? I'm fine with either. If I see raise your hand, I will call on you. If not, I will read it out. Okay, maybe I'll read it out then. So it's from Natalie Monacci. I may be brutally pronouncing your surname, apologies for that, who's got a question for Kristina from the University of Alaska, and she's at the University of Alaska Fairbanks. She asks, is MEAPAR currently working with or has plans to collaborate with the Alaska Ocean Observing System, the Alaska OA network, or the Atlantic equivalent? Maybe your North Pacific and Arctic neighbors attend the forum in March. Kristina.
Thank you so much, Natalie, for those questions. And yeah, I think the answer is hopefully an enthusiastic yes, but very beginning stages. So our community of practice here in Canada has recently started joining the US ALCAN's, the Coastal Acidification Network meetings, in hopes of building some of these kind of cross-border collaborations and connections. So I think there's lots of opportunity and interest to form some of these across the border collaborations on both coasts with groups like the Alaska OA network, as well as NECAN. And hopefully as well, some of the GOA-ON North American Hub activities will help build some of those connections. So yes, open to collaboration. For the BC OAH Forum, the current intention is to focus primarily on BC and folks within BC, but from that, I'm sure there's going to be further opportunities. And if you're interested in learning more, please feel free to reach out to me. And so I'll put my email address in the chat as well. So it's coordinator@oceanacidification.ca.
Thanks for your thoughtful answer, Kristina. I would also add too that the Canadian OA community of practice, we do try and let our members know of other activities that might be going on, that might be linked to, or have relevance for people in Canada. So if any of you are hosting meetings and you think that they would benefit from a kind of information exchange, then please do send Kristina a message and we'll happily post it on the website and let our members know about it and hopefully increase that cross-border collaboration. Thanks so much. We also have another question from Tanya Lopez, and this is for from Tanya Lopez, apologies, for Dr. Leticia. She says, great presentation, thanks. To the best of your great knowledge, what would be the most vulnerable region within the Mexican Pacific in terms of ocean acidification? And can you please point out a recent paper or reference around this topic?
Hi, this is a good topic for us because there are few information in the Pacific tropical ocean. So now this is a great opportunity to participate in 3 cruises to understand the knowledge for the ocean acidification and in this part. The most important contribution for this work is a division of the area, the latitudinal area, and it is a consequence for the carbon system ethnologically.
Great. And I mean, I think that also kind of links in with some of the observations like latitudinal differences that Kate has seen too. If there's a reference that you think would be great to add, please do just drop it into those questions and answer box there. There's a question from Dick Feely for Kate. What do you think is the reason that the southern regions have more corrosive volumes than the northern regions? Is it due to longer retention times or with more respiration? That's for Kate. Oh, sorry, that one was for Kate,
Okay.
although it's probably applicable for Mexico too.
Okay, it's important, the good question, but now we can make a more research for identify what kind of the part of this is the ocean assimilation or respiration. Is this in this moment I can answer directly the question.
Yeah, so it's a good question for more research. What do you think, Kate?
Yes, thank you.
Yeah. My thought on comparing the southern and northern regions is that my take is it's the longer retention time, but that builds the respiration signal. So it's definitely a combination of the two. And then as you then approach, like as you recall, when you approach the Columbia River, I think it's a complication of having the Columbia River presence as well.
Yeah, if I could ask a kind of follow-up question on that. I noticed like on slide 5, you had that kind of in the surface water figure that you kind of had a period of like corrosive water that was at latitude 46, so that's close to the Columbia River. And I just wondered that, is that directly from inputs from the Columbia River that kind of shows up? Has that been kind of attributed or?
I'm muted, so I'm sure you understood my answer. The Columbia River has a big impact on just the biogeochemistry on the shelf, and it can cap upwelling as well, so it can change how the upwelling is. It gets complicated because we move the river plume around with wind, but the Columbia River can make acidification more complicated and worse. And then it can also change how much water we're upwelling to the shelf.
Yeah. So live, a question for you, Kate. So just kind of continue on that. So you have live ocean over on the West Coast. Is it also on the Atlantic regions as well, or just a Pacific effort?
No, we cover British Columbia, Washington, Oregon coastal waters, and the Salish Sea.
Okay.
Yeah.
Okay, so there's not the equivalent. But you mentioned about the data associated with that. Is it easy to find where we can get this data from for anybody who might be working in those regions?
Yeah, and I can put it down in the chat. We can make our data available. We have some data up online, and then you can contact me for specific data requests. And we also make our forecast available on the NANU site. And all of those things you can get to. I'm going to post it in the chat. I can only post to hosts and panelists.
Okay, I'm sure Carly will make the magic happen.
Yeah, I'll forward it over to everybody else.
Thank you.
Thanks.
That's amazing. I mean, completely, so useful, and especially thinking about, the fact that that's very kind of complicated model outputs, that, not everybody has the capacity to be able to do that kind of thing. So definitely kind of facilitating that knowledge exchange is really, really important for the community. Does anybody else have a question? I can't see a hand raised. If not, I will happily ask questions because I've been so kind of stimulated by this discussion. I've got a question for you, Kristina. So one of the things you did was that you shared a kind of pathway for ocean acidification action. And I kind of got a question that's probably like a magical question of like, if you weren't constrained by funding or political will or anything like that, what would you think would be the most important action for kind of Canada moving forward on ocean acidification?
That's a great question. So politics, funding, everything aside, I think the most important thing would be the development of a centralized federal ocean acidification action plan that could be sort of like a linchpin to help coordinate and move forward actions. Because yeah, currently there's all these small efforts across the country and it's such a big area. So really having that sort of centralized mechanism for a reference that can be used, yeah, to kind of drive the needle or drive the engine forward. But what I could ask the same question to you, but
I know that's the things that kind of keep me up at night, you know, like in the magical
yeah.
scenario, like what should we be aiming for? You know, that kind of question. Great answer though. I've also, does anybody else have a question? Because if not, I'm going to go through my list. Please, this is a very friendly community. We're all friends here together. Oh, great. We've got a question from Shelley Walther online. Yes. Kate, she says, this is a question for you, Kate. Thanks for your enlightening presentation. Do you also share your model validation data and analysis? And do these include sensitivity analyses? Have you done work on biological impacts?
We share our we share the model validation data and analyses when we usually publish papers. So when we're publishing papers, the model validation and analyses go in those papers. We're happy to share when working on projects. If you want to send us your data, we will do the model data comparisons. That happened after I gave a talk recently at a WOAC symposium. Someone asked if I could compare our model data with their observations, and I did, and I just shared them because I was interested. Do these include sensitivity analyses? Sensitivity analyses meaning like when you change certain things in the model or to a creature? Yes. Which one?
I'll give Shelley the ability to come off mute. So Shelley, you're able to.
Yeah, so one really cool piece of work that's being done right now is a PhD candidate, Aurora Leeson, is looking, we have 100 wastewater treatment plants, and she's looking at the impact of turning them on and off in the model. So that's an example of when we're doing, like, to look at what happens when we change things. What's another example?
How about the things like phytoplankton growth rate or sinking or things that have to do with the phytoplankton?
Yep. Parker McCready has been looking at more like the sinking of CDOM in the model, and he's given a few talks on it. I don't know if he's published the data. He gave an ocean sciences talk about it. And we just submitted a proposal to relook at our like grazing rates and different rates for the phytoplankton community in the model. And a lot of these sensitivity analyses that we do are spurred by observation. So for instance, we just went through a round of model improvements where the Salish Sea, we needed to improve our burial rates in the Salish Sea for different nutrients and carbon. And we realized that we needed to fine tune that because we compared to observation. So a lot of times the sensitivity analyses are spurred by this one spot, the observations and models are off, and that's what motivates us to then do a model improvement.
I love it. Thank you.
Then you have you done work on biological impacts? I am a physical oceanographer and an engineer, and I love collaborating with people who know more about biology. So I'm really good at analyzing data and synthesizing information. And I don't exactly know all the ins and outs of biological oceanography. So that's more something that I collaborate with people on, or we work on working groups and provide our data. And we do a lot of like the data analysis, and we really rely on other people telling us what's important for that organism or for that food web, and we kind of just then work with them.
Thank you.
Well, the questions are coming in thick and fast for you, Kate. So we've got another question from Dick, which says, is it possible to show how retention time varies over the region in space and time?
Yes. Yes, it is possible. It would vary in space and time, and that's a really big question. Yes, we could either do through, processes. So it would be, processes that could happen, like what happens when there's higher river flow or different wind conditions. We could look at like how that process-based thing would change retention time. Or we could do something like, so that would be like a process-based study, or you can do something like where you release particle tracker, like little particles, and you see like where they're retained within the model. Either one of those are like a just quick way of that we could get at that question. And that has been done people have looked at retention time and how it changes using live ocean model data. And then also Jillian, who was a postdoc in the coastal modeling group, she also compared different particle tracking methods to like get at questions looking at retention time. So she compared different ways the model would track particles, and she published a really nice paper on that.
Nice. Another question for you, Kate. You're not out the hot seat yet.
Okay.
Question from Anna Hughes. Do you incorporate benthic data, so oxygen, DIC, nutrient fluxes, into the model as well? And additionally, does the model investigate hypoxia, or is the main focus on OA?
Great question. The main focus is not OA. It was just the focus of my talk because it's OA week. But we do focus, we work on hypoxia, and we have the models used to understand productivity dynamics. It simulates physics and biogeochemistry, and any way that we can try to get the model to be used to answer questions in our region, we try to get that to happen. So definitely yes, OA and hypoxia. Do we incorporate benthic data? Yes, and if you have additional benthic data, please send it to us. We're always looking for more benthic data. A lot of our rates come from the RISE project on the shelf, and we then try to piece that together with data that's in the Salish Sea on benthic rates. And I think that there is some work now that they're trying to connect, get more sediment oxygen demand rates within the Salish Sea. I think people out of Western Washington are trying to do that. Sam Kastner and David Shull, I think, are trying to look at more data for that.
Great, thanks, Kate. We've got a question this time for Leticia, which is coming from Jan. She says, great talks, all of you. Leticia, what is your biggest data gap? That is, what data do you wish you had? And she says, gracias.
Sorry, we can repeat the question because
of course
the Wi-Fi is yes, please.
Yeah. So the first part of the question is, what is your biggest data gap? So which data do you wish that you had?
The statistical what I use it is as a first one is as a cross correlation statistical. Also, we use the Bayesian statistical analysis to compare the old regional data with the data with the 2018 2019. So in this we have a statistical significance difference between years and between zones with this statistical.
Right. I was noticing that it was a nice example of international collaboration, that on some of the papers that you showed, you had US authors on there. And you know, I was thinking about the work that you're building up in terms of your knowledge on anomalies that may be happening, and you know, these neutral years and things like that. I was just wondering whether you had any plans for maybe like an international cruise in 2026 or 2027 to see if that will also be an anomalous or a neutral year.
This is a good question because this is let me explain. All the support, financial support from Mexico is limited now. So the only research vessel that they can use is the IMIPAS research vessel, but this research vessel is to move it to the Atlantic to the Sargasso investigations. So in this case, we don't have any opportunity more in Mexico, but if open to sharing this information, and if the other countries invited us, we're happy to participate. Yes.
Okay, it's good to know. I feel like there's a little bit of a move internationally towards less investment in science, which you know, when we're working on these big questions like this that need these very large legacy data sets, it unfortunately limits what we can do. But yeah, maybe the community can come together and make some magic happen. Yeah, fingers crossed for you. We also have another question from Shelley, and this one's for Kate again. So Kate, are the CTD data from NPDES wastewater permittees included in your model validation analysis? And then the second part is, if that is so, have you found the O2, pH and or chlorophyll A measurements match well?
Yeah, that's a great question. I personally have not compared any data from a NPDES permit result, so like any data with the model. But dischargers, that comparison will be more done in Aurora Leeson's work, and our group, the UW's coastal modeling group, is looking at turning on and off the wastewater treatment plants. And also Susan Allen's group at UBC is looking at a similar thing. So they're using two models to then see this, if the results work for King County. And out of that work, there will be more model data validation between dischargers and models. And that has not been something I have personally done, so I can't really comment on that. I don't do that. I'm looking at more like mooring data and cruise data and bottle data from different sources.
Shelley says, thank you. Does anybody else have any more questions? I mean, it's rare that you get to have these direct question and answer sessions with three such great presenters representing different areas. If there isn't going to be any more questions, I might pass it back to Carly.
Sounds good. Thank you, Helen. And thank you to all of our speakers today. I'm just going to share my screen one last time. Yes, and last but not least, I'd like to thank all of you in the audience for joining us for OA Week 2025. Please don't forget to consult our website via the QR code on the left-hand side of the screen here, and you can register for any other sessions in OA Week that may interest you. We have 2 days left of this wonderful symposium, so make the most out of it while we're all here and gathering together online. We'd especially like to highlight our neighboring hub, LAOCA, whose session is on Friday. If you'd like to stay up to date with the GOA-ON community, consider signing up as a GOA-ON member, and you can do that through the QR code on the right-hand side of the screen. All right, and that's all we have for today's session. Thank you all so much for joining, and to our speakers for their presentations, and see you next time.
Thank you to all our speakers and everyone for joining. Great work, everyone.
Bye, thank you.
Bye, everyone.